Hello friends, welcome back in the channel of Clinical Care Insight. We started a series of lecture on the introduction to microbiology. Until now we studied about what is the meaning of microbiology, okay, what are the scopes, as well as what are the, you can say, the subfield of microbiology, okay. Then we also seen about the different you can say the kind of research which is happened in the microbiology. We refer as a discovery, and we seen the discovery era. We see also the golden age in the microbiology, okay. And we are left with the one topic called as the microscopes. So in today's lecture we are going to discuss about the microscope. I hope you all are fine. I am also fine, okay. Now see the lecture outline. Today we will talk about the microscopy introduction first, then their types. There are different types of microscopes, okay. Then the general structure of the microscope, and also about the heading of microscope. So make the heading microscopy. Microscopy. Microscopy is basically a field of study where we are going to use the microscope in order to see the smallest thing, It is not only limited to seeing the only about the microbes, okay, only about the pathogen, you can say. It is also used in different fields, in different industry. But for our discussion, we are discussing those microscopes which are involved in the microbiology, okay, which are involved in basically understanding about the microbe structure. Fine. So microscopy is basically a field, okay. It's basically a technical field where we will use the microscope. So microscopy is a technical field, okay, where we are using the microscope. Got it? No problem. Now you know that microscopes are you can see the microbes are so small that we cannot see by our naked eye. For seeing their structure, for seeing how their, you can say, the life cycle is working, okay, how their life cycle is, in which pattern and they are producing their, you can say, they enhancing their generation, how they are replicating. So all these phenomena will only seen under the microscope, okay, under the microscope. We also discussed when we are describing the history of in the microbiology, we seen that the first microscope is discovered by the Galileo, okay. Galileo Galilei first discovered. Then we seen that there is advancement in the microbiology, and you can say there is a discovery of one special kind of microscope called as the electron microscope, discovered by the Ernst Röska. Okay, we've seen. Now, once we understood about the microscopy, and one thing you should clear in your mind, there are different types of microscope. Then question arises, why there is a need for different type of microscope? The answer is simple. Sometimes we have to understand the microbes in a superficial way, okay, and sometimes we have suppose if we are doing the research, okay, and we are involved in understanding the, you can say, the minute fine details of a bacterial cell, or you can say the eukaryotic cell as well. For that, understanding the inside environment of a cell, okay, or a living cell, we have to use the advanced microscope, okay, we have to use the advanced microscope. So for that, there are different types of microscope. So now let's deal with the first types of microscope. So if we discuss the type of microscopic types, then mainly microscope can be divided into the two classes. One is called as the optical microscope. Have you heard about these microscopes? Optical microscopy. Fine one. And the another class we have is the advanced microscopy, and advanced microscope called as the electron microscope. Electron microscopy. Fine. Now, what are the optical microscopes? Optical microscopes are those microscopes where we are using the different kind of lenses. Okay, we are using the different kind of optical lenses in order to see the microorganisms. Remember, see, the microbes are very small. Microbes are very small. So for seeing them, we have to magnify their structure. Okay, you clear about, first clear this two term called as the magnification. Magnification, and another one which is frequently used when you describe the microscope called as the resolution. Resolution. Fine. Now, magnification means enlarging the object. Okay, suppose this chalk actual size is this much. Fine, but in the microscope you can enlarge it 100 times, 200 times. Fine. So this enlarging and the object structure, okay, that is called as the magnification, and it is represented like 4x, okay, 2x. Probably you've seen about this. Now, the resolution means to understanding the fine detail of the object, to discriminating between the two closely related things. Okay, that is called as the resolution. So, if we talked about the optimum microscope, then optimum microscope must have the high magnification power as well as they must have the good resolution. Okay, they must have the good resolution as well as the magnification. So clear with these two terms? Fine. Now come to the optical microscope. As I am describing, the optical microscopes are those microscopes where the lenses are utilized. Okay, different optical power lens, they are utilized in order to see the microbes, in order to see the microscope. That's why these optical microscopes, they are also called as the light microscope. Fine, they are also called as the light microscope. No problem. Now come to the electron microscopy. This electron microscopy, or you can say the electron microscope which is used in the electron microscopy, they are the advanced type of microscope. They are the advanced type of microscope where the electrons are used to generate the three-dimensional, you can say, view of a specimen. Specimens means like suppose this is the blood. Suppose the blood is a specimen. Okay, the tissue is a specimen. Specimens means the object whom we are going to study on the microscope. That are referred as a specimen. Okay, so in electron microscopy, there is a utilization of electron beam by which there is a generation of 3D image. There is a generation of 3D image, or there is a scanning of the image. Okay, scanning of the specimen and see their detail. That is called as the electron microscopy. So you should write it down about the electron microscopy. First, it is an advanced microscopy. Advanced type of microscopy. Okay, another thing is here the electrons beam are used, okay, in order to generate the three-dimensional view. Fine, no problem. Now the optical microscopes are again the three types. Okay, it is again the three types. Okay, so you write it down about these three types. The first one is called as the bright field microscopy. Bright field microscopy. Okay. Now the another type is called as the fluorescence microscopy. Okay. Fluorescence microscopy. Fine. And another one is called as the phase contrast microscopy. is contrast microscopy. Now, these there are the three different types of optical microscope. The first one is the bright field microscopy. Fine. Another one is called as the fluorescence microscopy. Okay, and the next one is called as the phase contrast microscopy. Now, these three different microscope have the different purpose. Fine. This one, the bright field microscopy, they are frequently used to study about the blood. Okay, to study about the tissue. Okay, the tissue structure and the study of the blood. We are utilizing the bright field microscopy in the lab. Okay, so you should write it down. When we are using the bright field microscopy, we are using it first frequently. It is frequently used microscope in the laboratory setting. Okay, now the second thing is it study the blood smear. Okay, and it is also used in the study of tissue structure. Fine, yeah, no problem. Now, this fluorescence microscopy, they are utilized in the study of immunofluorescence, like antibody-antigen reaction. You probably heard about the ELISA test. Okay, so fluorescence microscopy means you can say basically observing the living tissue without the staining. Okay, without the staining. This microscope have you can say capacity to, you can say, generate one kind of you can say the light, okay, the fluorescence, and by which the transparent object will show the color. Okay, so here in the bright field microscope, you have to you can say stain the microbes. You have to utilize the different types of stains, different type of chemical. You have to basically color the microbes in order to see. But in the case of fluorescence, there is no use of staining. Okay, no use of staining. Fine, and this is commonly utilized. Okay, this is commonly utilized for studying the immunofluorescence studies to detect the specific antigen in the tissue, and it is also used in the fluorescence, you can say, level, okay, antibodies, fluorescence level antibodies. No problem, yeah. So you should write down the example. Okay, it is used in the study of immunofluorescence. Immunofluorescence studies of what? Antibody and antigen. Okay, no problem. Now come to the phase contrast microscopy. This phase contrast microscopy, we are using it also without, you can say, staining the, without staining the specimen. We are using this for understanding the cell, living cell, okay, and the tissue. Here in the phase contrast microscopy, what we are studying without staining first, without staining. Okay, and another speciality of phase contrast microscopy is, by the help of phase contrast microscopy, we can see the living cell. We can see the living cell. No problem. Now let's switch to the electron microscope. Electron microscopes are the two main types. Okay, electron microscope are mainly the two types. The first type is called as the scanning. First type is called as the scanning. First type, the electron microscope is divided into the two parts. The first type is called as the scanning electron microscope. Electron microscope. Fine. Now, and another one is called as the transmission electron microscope. Okay, the next one is called as the transmission electron microscopy. of microscope. Now, in short, it is called as the SEM, okay, whereas the transition electron microscope, they are called as the TEM. Now, these both serve for the different purpose, both serve for the different purpose. Now, in the scanning electron microscope, we, by using the electron beam, okay, the laboratorician generate the three-dimensional view of object, or we can say the three-dimensional view of a specimen. Okay, it is the specialty of scanning electron microscope. Whereas in the case of the transmission electron microscope, we are studying the ultrastructure of cell. Now, what is mean by the ultrastructure? First, you should write it down. Ultrastructure of cell. Okay, ultrastructure of the cell. Now, see, we seen that in the you can say the optical microscope, we sometimes we have to stain the specimen, or sometimes, like in the case of phase contrast microscopy, we don't need to stain. Rather, there is a, you can say, changing the contrast of a specimen. Okay, the transparent specimens will, you can say, illuminate, will show the color by when we using this phase contrast microscope, because it holds the special kind of lens. It holds the special kind of lens. And the thing is, we are only see superficially the, you can say, the specimen. We are not, these microscopes are not able to understand the internal structure, which is called as the ultrastructure. Okay, inside the cell content is called as the cell organelles. Fine. These cell organelles represent the ultrastructure view of cell, which is only you can see while we are using the transmission electron microscopy. Here, what is happening? Through the electron beam, there is a generation of the three-dimensional view of object. No problem. Now, let's summarize it. What we studied. We studied that the microscope, there are different types of, first of the microscope, but mainly we can categorize the microscope into the two basic form. The first one is called as the optical microscope, also called as the light microscope. Here we are using the different diopter of the lenses, different power of the lenses, and these lenses is basically, you can say, this lens basically have the high magnification power, which is the required one, and another one is the have the resolution. The magnification means enlarging the object. Okay, enlarging the object, like it may be 4 times or 2 times from their actual size. No problem. Now, resolution means the 2-point discrimination. Okay, through the help of resolution, you can understand the things. Okay, you can just separate the very minute things. Okay, no problem. Now, let's come to the division. The optical microscopes are divided into the 3 types. The first one is called as the bright field microscopy. In the bright field microscopy, and this one is a very frequent type of microscope in the laboratory setting. Okay, now here we are, you can say, checking the blood smear, okay, and also studying about the tissue histology. Okay, we can say the tissue topography. Okay, the tissue topography means tissue arrangement. Now, in the fluorescence microscopy, what is happening here? Basically, we will study, or we can say basically we will labeled the cell with the fluorescence material. Okay, and then we will observe in the microscope. The one of the most frequent used, you can say, the part is when you labeled the antibody in order to understand about which type of antigen is present in the specimen. Then on that time you are basically labeled those antibody, and when there is a reaction, then that reaction will clearly you can say the visible under this fluorescence microscope. Now, in the phase contrast microscope, there is no need for staining. Okay, there is no need for staining, and you can directly see the living thing. Now, the another major class of microscope is the electron microscope, which is the advanced microscope, and comparatively it is a larger one. Okay, it is a large type of microscope. Now, here And don't forget that this is discovered by which we already discussed in our previous class, Ernst Ruska. Okay, Ernst Ruska is discovered the electron microscope. Now it is an advanced type of microscope, and here we are using the electron beam. There are two principal type of the electron microscope. The one is called as the scanning electron microscope, where there is a generation of 3D view of the specimen. Okay, now in the case of transmission electron microscope, here we are seeing the ultra structure of cell organelles. We are seeing the sometimes we have to, and this is mainly utilized in the research field. Okay, they are mainly utilized in the research fields like the bacteriology, virology, parasitology. Okay, all those you can see the branches of the microscope. We are using the transmission electron microscopy. No problem. Now, once we understood about the types, let's move ahead and discuss the general structure of microscope. Because these all microscopes you can see in a better way when you seeing there in you can say on the pictorial form. Okay, if you see the pictures of this microscope, you will understand it in a better way. So now let's move ahead and discuss the general structure of the microscope and their parts.